Okay, so actually today's lecture was much longer. So last year we had 3 hours for it, but this year we only have two. So I will concentrate even more on the important ideas. So today, what we will see are different mathematical models that can help us to model this network of agents that are interacting with each other. So uh, we will start with these static network models. So this is just a beautiful picture to illustrate the idea. So we will have different agents and they will relate with each other. And then how do we analyze these relationships? So you have these are at all different levels. So you have it here for instance with genes, with proteins. You have it with cells, with individuals. So these relationships and these graph representations of the relationships, they are ubiquitous. You can find them at all levels of biology. And here you have a database where you can download or explore relationships at the level of protein-protein interactions. And it would be useful that you enter into these links and you explore them a bit. They are very interesting. And then there are all these representations, they are based on graphs. So a graph is a couple of sets. So one set of nodes that are these V1, V2, V3, and a set of edges. So a graph is a set of nodes V and a set of edges E. And the edges can be directed. So from node 2 you go to node 1, but you cannot go back. Or they can be undirected. So simply these two nodes are related and they are related in both directions. And then you can put labels to the edges, and these labels can be relationships. So for instance, V1 teaches V4 and V2 is the team leader of V4 and so on. Or these labels can be numbers. So a matrix, a graph is represented by an adjacency matrix. So the adjacency matrix, you put all nodes in both axes, and then you put a 1 if there is a relationship from V1 to V4. So for instance, here, there is this edge. So this edge starts at V1. So the columns or the rows are where the relationship starts, and this is where the relationship ends. So this one is starting in V1 and finishing in V4. And this is an a directed graph, so there is no connection between V4 and V1. Okay, so this adjacency matrix is symmetric if it is undirected. So undirected, you see you have V1, V4, but you also have V4, V1. So this adjacency matrix is symmetric. So if you transpose it, you have the same matrix. And here you simply put ones, here you put the weights. Okay, so then you can define a lot of properties of graphs. So for instance, you can count how many connections a node has. That is called the degree. So V1 is connected to V2 and V4, so it has degree 2. V2 is connected to 3 nodes, so it has degree 3, and so on. You can also define paths. So a path is a path, is a sequence of vertices that are connected by edges. You can also define cycles. So for instance, here you have a path that is closed. So you start at 3, then 2, then 4, and then 3 again. So in a cycle, it is a path that starts and finishes in the same vertex. Then you can define connectedness. So a graph is said to be connected if there is a path between any two vertices. So this this graph, the green one is connected because between any two vertices, there is a path. But this one, if you consider all the nodes at the same time, then the graph is disconnected. Okay, so you cannot go from V8 to V2. There is no way to go in that way. And then planar. A graph is planar if it can be drawn on a plane without crossing edges. So here it is a non-planar. There is no way to draw this graph in a way such that the edges are not crossing. And this is a planar graph. And this is a bipartite graph in which you have vertices, but the vertices belong to two different sets. So let's say that these are genes, these are proteins, and somehow these are relationships between these genes and these proteins. So you have vertices, but the vertices belong to different sets, to different natures. Then these kind of graph representations, they appear in system biology. This is a very well-known paper of system biology. This guy, Yuri Allen, has a very famous course on system biology too. And it discusses that there are graph patterns that appear over and over in many different setups. And for instance, there is this gene regulation sub-pattern. So X controls Y and Y controls Z, but then there is an extra control X to Z. Okay, so this pattern is called the feed-forward loop. And this happens with neurons, this happens with food webs, so with relationship between the species. And this pattern also appears in many at different levels, so at genes, at neurons, with food webs, and so on. So these are, so given a graph, you might be interested in subparts of the graph that are repeated. Okay, so this paper discusses these repetitions, these subpatterns. Okay, let's go to here. So one of the things that you can define are quantitative measures of graphs. So quantitative means given a node or given a graph, we will make some calculation and come with a number. So for instance, here I can, so this is, I'm counting the number of neighbors of V, that I will call it KV, and then I will define the clustering of a node. So V is a node. So the clustering of a node will be a number. And if the number of neighbors is smaller or equal 1, then it will be 0. And otherwise it is this number. So this number is 2 times a number that we haven't defined yet, divided by KV times KV minus 1. So except for this TV that we haven't defined yet, everything should be clear up to here. Okay, so what is TV? So TV is this thing. Okay, so let's see. So I'd like to copy so that we can see it more slowly. So we have TV is equals that. Let me check. UW. And this is, yeah. That's that. UW. And UW. UW equals to H. Okay, so let's start. So first we have a set. This means that we have a set. And this means that we are counting. So this is the cardinal. So we are counting how many elements are in this set. Okay. So the set of what? So the set of we have a node, and then we have the neighbors of that node, and U and V are neighbors of that node. Okay, so let's draw that. So we have V, and then we have U and W. They have to be neighbors of V, so that there must be some connection here, because otherwise they wouldn't be neighbors of V. So we are looking for U and W that must be nodes. They must be connected to V, and UW must be in the set of E. So it must be an edge. So let's make this red. So there must be some connection between U and W so that this is in E. Then we are counting. So this node has many other neighbors. So it has many other neighbors here. It's connected to many other things. And then we are counting how many edges are there between the neighbors. So that is this TB is for the neighbors of V, count how many of them are connected. Okay? And then we will define the clustering of the node V as twice the number of connections between neighbors divided by the degree or number of neighbors of V, that is the degree of V. More clear now? Yep. I think it is very important to learn to read this kind of definitions. Okay, next. So there are many different types of graphs. So for instance, let's say that we count, I'm here showing only two of these three kind of graphs. So let's say that we have nodes and we have randomly random connections between all the nodes. So this is a kind of graph. A different kind of graph would be locally the nodes are very much connected. So locally they are connected. And then from time to time you have a long distance connection. Okay. So from time to time you have these long distance connections. And then this kind of graphs they are called a small world. Or you could have something that are called scale-free networks, where a few nodes, you see a kind of hierarchy. So for instance, all these nodes they are only connected to a higher, so this would be low hierarchy. This would be a little bit higher in the hierarchy. So it is connecting the lower node. And then there is a higher, even a higher. So you have a few nodes that are gathering information from the low level. So you have these three that are gathering this information, and then all the information is gathered into a central node. So if you analyze what is the number of relationships of each one of the nodes, and you plot here number of relationships and number of nodes with that number of relationships, you have that the different types of graphs, they behave differently. So for instance, this one here, this average distribution is associated to these random connections, while these scale-free graphs, they behave like this. So there are a lot of nodes with very few connections, and then there are a few of them with a huge number of connections. And this kind of relationship, this is a Gaussian, this is a power law. Power law is number of relationships to some negative power. Yeah. So for instance, friendship relationships. So you have among your friends, most of your friends know each other, so they have local connections. So there are a lot of local connections, but then there are two groups of friends, and some of here has a connection with this one. So friendship relationships, typically they behave as a small world. And scale free is an example of computer networks, or even the telephone networks. So the telephone networks, all of these would be the smartphones, and then the smartphones are connected to the local antenna, and then the local antennas are connected to higher elements and higher nodes in the hierarchy. So when you phone from here to there, somehow your information has to travel through the higher hierarchy nodes. So different networks, they are set up in different ways. Okay, so yeah, this is a representation of the same idea. So this is random, a small world, and scale-free networks. And then if you count the distribution, you see that this is kind of Gaussian, this is also kind of Gaussian, and this is a power node. And here you have a database of networks of proteins. And again, it is very interesting if you enter into the database and explore a bit, especially for your project this year. This year is, if I have a disease, which are the proteins involved in that disease? That could be targets of some drugs. And these networks help you to identify which possible proteins, how proteins relate to each other. So if you know that a protein is involved in the disease, probably the proteins in the neighborhood, they can also be targeted. Okay, and another web with networks of proteins, and yeah, so databases that record this kind of information. More databases, you can explore each one of them. More databases, so that there is really a lot of information about this kind of stuff. Yeah, and many of them, so for instance, and they have all these databases, they have different focuses. So for instance, this is gene co-expression networks. So genes that are expressed at the same time. So the nodes are the genes, and if they are expressed at the same time, they have an edge. Or gene regulatory network. So if this gene regulates this one, then there is an edge, and so on. You have a relationship between genes and diseases. So this is actually for your project this year. Gene and drugs, yeah, many other things. Okay, then an interesting set of networks is these Bayesian networks. So Bayesian networks is not only that the elements are related, but also that we can compute probabilities associated to these relationships. So for instance, let's say that we have decided relationships. So for instance, this is a very classical example in which you have a house and you are not at home, and then the alarm of the house sounds, and it can sound for two reasons. First, because someone entered into your house, or because there has been an earthquake. And then the alarm is sounding, and these are two neighbors that know that you are not at home, and then they call you. And you associate different probabilities to each one of these events. So having a burglary, it is a relative rare event. So only 0.2% of the cases of all the hours of the year, only 0.2% of those hours there is a burglary. And the same for earthquake. And then most of the time there is no burglary, there is no earthquake. And then depending on whether there is a burglary or an earthquake, so here both of them are active, then what is the probability that the alarm is sounding, and then what is the probability that the alarm is not sounding? And then you have all these probabilities, and once the alarm is sounding, what is the probability that David calls you, or the probability that David does not call you? So if the alarm is sounding and if the alarm is not sounding, and you have these two probabilities, and the same for Sophia. And now you have a call from both Sophia and David. The alarm was sounding, so you can calculate what is the probability that all this event happen. So Sophia is calling me, that David is calling me, the alarm is sounding, but there has not been any burglary, and there has not been any earthquake. Actually, there is a mistake in this slide. I corrected in my slides. So the one we should be using is this one. So you have to substitute the 0.001 by 0.999. Okay, so you can decompose the probability of this event according to this graph. So according to the graph, the probability of Sophia calling you, so Sophia calling you, depends on the alarm. So probability of Sophia given the alarm, and the same for David. And then the alarm depends on two things. Then these two things is burglary and earthquake. So probability of alarm, and then I'm interested in this thing. So not burglary, not earthquake. And then the probability of not burglary, probability of not earthquake. So, and now you can put numbers on that. So this, 0.75, this, 0.91. This is this mistake. You see, I should be reading this cell, not this one. So 0.999, and probability of not B, probability of not B is 0.998, and you have all the numbers, and then you multiply, the true result is 0.67, something like that. So, but the interesting thing is that now we don't only have a graph of relationships, but we can also associate probabilities to these events. And this is very much used also in proteins. And so let's say that you have a cascade here of signaling, and this cascade depends on two things. That is, these two receptors that you have in the membrane being bound, and then you have the different probabilities. The probabilities of this receptor being occupied, the probability of this receptor being occupied, and then the probability of this cascading happening. So probability of cascading given that R and S are occupied, probability of cascade only if only R is occupied, if only S is occupied, or that both of them are not occupied, but C is spontaneously elicited, so it is not spontaneously used. And then you have for another protein, you have or another cascade or whatever, you have same kind of probabilities, and then you're interested in what is happening with this gene G, and then you want to see if it is expressed or not. If the cascade is active, then the probability of G being expressed is very high. If C is not activated, then the probability of G is very low. And then you have to estimate. So given the probabilities, it is very easy to propagate the probabilities. How do we estimate the probabilities? And then here you have the solution. So you have to measure, and the measure is you take a snapshot of cells, and then you measure, is R occupied, is S occupied, and what is C? Is it active or not? What is G? And what is set? So you have a lot of snapshots of cells, and then for each one of them, you measure all these activation states. And then from these activation states, somehow there are algorithms that tell you how to compute these probabilities. And once you have the probabilities, you can easily simulate what will happen if this receptor is occupied. So, and in general models, you also have these two uses. One is to simulate what is happening, but to simulate what is happening, I need the parameters of the model, and then the parameters of the model, I need data, and then I fit the data to the model. I fit the model to the data, sorry. Okay, and here you have libraries or programs that can that allow you to do to handle Bayesian networks. And a different kind of networks is these Boolean networks. Boolean networks is somehow an evolution or a different approach to networks, which you have still the network relationship, but the relationships are like these logic functions. So, for instance, we can express how the nodes, how the states of the nodes will evolve over time. So you can have this, for instance, let's say that you are looking at 3 nodes, whatever. So it is the activation layer, the activation state of 3 proteins, for instance, or 3 genes, and then none of them is activated. If none of them is activated at time t, the probability of being activated in t plus 1 will be 80%, and 20% of the cases they go to 1, 0, 0. So meaning that the first one is activated and the other 2 are not, and then you have the different probabilities of moving from one to the other. Okay, so these kind of networks, and you can put also equations on that. So V1 at t plus 1 will have the same state as V3 with probability 0.8, and then V1 at t plus 1 will have the negative of V3 with probability 0.2. So you define these probabilities, and then again, it is as we had before. So we have 2 uses of the model. Always you have 2 uses of the model. One is to simulate what will happen. So then we can try to imagine paths along this graph. The other one is, given the data, how do I estimate these probabilities and these relationships? Okay, so how do I estimate the model? This is the model. How do I estimate the model from the data that I have? And there are algorithms for both. The simulation is very easy. The estimation of the model is much more complicated. And then here you have different programs and libraries to work with these Boolean networks. And there are networks at all kinds of levels. We are mostly concerned in this course with the cell and molecular networks. And then you have signaling networks, transcription networks, protein-protein interaction networks, genomic networks. And for each one of these networks, you can put the models that we have explained. So we can put a plane graph, we can put a Bayesian network or a Boolean network. Yeah.
In the probabilistic Boolean networks, how is it possible that the node V1 has the same state that V3 and the negative V3?
These 2?
Yes. How is it possible that it can be in the same state, that is a state and the
So V3 will cause V1 to V3 will have an influence in V1. And this influence is V1 will have the same state as V3 at t plus 1, or the opposite. And then this effect is much more likely than this one.
So V1 can have either of the 2. Okay.
Yeah. And most of the events in biology, they are random. So it is not, you have seen, we saw it the other day with the dissociation and association of molecules. So they happen at random. And that random nature propagates all over at all time scales. So if you have an active gene, this active gene is activated, and normally it has an effect in this other gene, but it doesn't mean that it will be immediately and with 100% of efficiency and so on. So there is some randomness in the process. And another way of analyzing this kind of relationships is with these metabolic networks. For metabolic networks, let's say that we have three chemical reactions. So we have, for instance, CP or PC, which is a isomerization of a molecule, or you have C and P that combines into CP, or CP dissociates into its two components, and then same here, another chemical reaction. And then another approach that is also very much used in system biology is to try to describe how the concentrations of these 6 species, if you count how many different kind of molecules we have there, we have 6. So how the concentration of these 6 species will evolve over time. So for a closed system means that there is no external input. The relationship between them will simply be related to the concentration of X and the velocities associated to those concentrations. So let's see in this example. So in this example, we have 6 species. So we will have 6 rows in this matrix, and each one of these reactions will give you velocities. So these velocities, for instance, here x1 is converted into x2 at the, so this is reaction 1, reaction 2, reaction 3. So in the right direction, the velocity of how much PC or how much x2 is formed depends on how much x1 you have. If you have very little x1, then you cannot form too much x2. And if you have a high level of x1, then you can form much more x2. So it is a constant times how much x1 you have. And in the opposite direction, it is another constant on how much x2 you have. Then, for instance, the second reaction, you have a constant to the right, and you need x3 and x4 to encounter each other. So we have this kind of relationships where we have x3 times x4, and this multiplication accounts for the encountering of the two kind of molecules, and then x1 is decomposing at this rate, and the same for the B3. So now you see that this is a vector of time of size 3 times 1. This is 6 times 3. So this is well defined. This is 6 times 3 times 3 times 1. So overall, it is 6 times 1. That is what we need for the derivation for the derivative. So this differential is how is the rate of each one of these concentrations changing over time, and is defined by the velocities of the different chemical reactions. So again, if you measure these constants, then you can very easily simulate what will happen with this system. And you have that, and again, at all different kind of levels. Probably you're familiar with this kind of metabolic reactions. So these metabolic reactions you have, this is exactly the same as we had before. So you have a number of species, and you have some ATP and ADP is also some other species. So you have many chemical reactions. You can write how the concentration of each one of them is changing over time as a function of the velocities of the different reactions. And then you have to compute somehow what are those velocities. And sometimes it will be very simple models like this one, or some other times it will be more complicated models. Here you have a Michaelis-Menten. This one is a Michaelis-Menten velocity. So Michaelis-Menten is saturated. So you have something like this. So this is velocity, this is concentration, and then it behaves something like this. So at some point it saturates, cannot go faster, no matter how much X you have, cannot go faster. Typically, this is when you have an enzyme mediating some chemical reaction, you have this kind of relationships. So if you have parameters for that, and you have the initial conditions, then you can very easily simulate what will happen with this chemical, with all these species. And this is an example of that. So if you have the initial concentrations and you have the parameters, then you can easily say what will happen with each one of the species. Then, yeah, so then this is, let's say that we have an open system now. So an open system is not closed. We can have inputs from the exterior. We can also get rid of some or excrete some of these species. So now the new term that appears is this one. So with a closed system, it is this. With an open system, now you're communicating with the exterior. And this tells you, this differential equations tells you how the system will evolve over time. Let's say that you are at a steady state. So you are at a state where everything is constant. So if everything is constant, then there is no change of the chemical reactions of the concentrations over time. So if there is no change, that means that this term has to be zero because there is no change. And then this is much easier to solve because now we don't have a differential equation anymore. We have a standard equation now. And then it is simpler to solve. And this family of methods that analyze the systems at a steady state, they're called flux balance analysis. They're also very much used. And then here you have this guy, Bernard Paulson, is a well-famous, very good, well-known guy in this flux balance world. And here you have a paper in which he describes. But at the end, it is, you see, at the end, it is a set of equations that depend on the velocity equals zero. So it is a linear set of equations. And then you can try to derive, so if you have a set of cells that are at a steady state, then you can try to drive these cells towards a point that you like. For instance, this is used in bioreactors. In a bioreactor, you want the cells to produce maximally a given compound, and then you can play with the nutrients and the exterior. So the exterior is B. So you can play with B so that you drive X to be, or the velocities to be at a given point. So this is very interesting. Then toolboxes that allow you to work with these kind of models. Any questions so far? No? Okay, so then I think we can make a break here. Then let's continue. So now we will focus a little bit more on the mathematics of these systems. So we have introduced 3 different kind of models. First one is based on graphs. So graphs is just an interaction model. So there are no stoichiometry, no parameters. They're kind of static models. Maybe the Boolean networks are not so static, but otherwise they are simpler models. And then we have seen the other extreme of models, that is where you have a dynamic system, so you have the chemical reactions, and then the stoichiometry is involved, you have the kinetic parameters, and then you would have something in between, where you are lacking the kinetic parameters, and then you can only focus on fluxes. You will see that along the slides I'm referring to some papers. These papers are, in general, they are very informative. They are very general, very useful for you to read. And if you want to present them, you can do it. And then we will see now, we will start seeing different kind of approaches to mathematical models. And the first one will be these finite state machines. So finite state machines is a machine in which you have a vector or a state that is defined typically by a vector, and then this state changes from one over time. So for instance, here you have the state machine of the traffic light. So the state is red, and then it goes to green, and then eventually it will go to yellow and back to red. And so this kind of automata is what we will see. And you can define this automata based on some probabilities or some events. So for instance, you start zero here at the state 0, and then with probability B, you go back to 0, or with probability A, you go to 1, and then again with probabilities. So these A and B can be probabilities, or they can be conditions. So if you're at 0 and A happens, then you go to 1. And if you are in 1 and B happens, then you go back to 0. So these automata can be understood depending on what is your problem. You can define it in a probabilistic way, or you can define it in an event way. And then, for instance, these relationships of genes regulations, they are typically defined in these events. So let's say that you are the state of your system is defined by a few genes that you are looking at. And then if this gene is activated, then it represses this transcription factor. And then this transcription factor prevents this gene from being expressed. So it is an automata of this kind. And then you can express it at the level of automaton, like these ones, or you can, this one and this one is the same. So it is, this is a graphical representation, this is another graphical representation. So these bars, they mean that they prevent. So if U is activated, it prevents Y from being activated. And this one is blocked by U1 and U2, and this S1 blocks Y, but if A happens, then A blocks this inhibition. So, and then you can write that in terms of Boolean network equations. So you can say Y is always expressed, or Y at t plus 1 is the negative of U. It's the negative of U. Or Y at t plus 1 is the negative of U1 or U2, or you can write the logic of any one of these systems, or you can write even the differential equation. So it is not only that it prevents, but it prevents at which rate. And then here you have the rate at which they are prevented. And then, depending on how, which are your parameters and all that, then you can have, for instance, the behavior over time. So you start with, you write some equations of how A and B evolve over time, and then how these A and B is affecting genes that you are interested are 0 and 1, T A, T B, whatever. And the Boolean networks, they are 0 or 1 responses. So you evaluate at time equals 0, at time equals 1, at time equals 2. You follow these equations, and then the equations in the paper, I mean, not these ones. But we follow the equations in the paper, and then you would see that it behaves as a 0 or 1 responses, and you have the 0 or 1 responses. The differential equations, what gives you is a refinement of this simulation. So it tells you, okay, this one is activated, but depending on what is the rate of activation or inactivation, you see that the response is not completely sharp, but it has some delay to go on and to go off. Then another family of models are these recursive deterministic models. So recursive deterministic is you have some state, and then the next state will be a function of that state. And here, for instance, it was the paper was analyzing what was the yeast cell cycle, and what it saw is that, well, it defined a few genes that were of their interest, and then they defined a network in which each one was influencing other genes, and then they defined the influence. So the influence is when a gene is activated. So when this is activated in the next state, this one will also be activated, or it will be repressed. So when this is activated, this one is repressing this one, and this one is repressing this one. So the red arrows are inhibitions, the green arrows are activations. And then the function, so the state is defined as 0, 1, or the previous state, and 0, 1. So you have these arrows, they take values 1 or -1. The states of each one of the genes is 0, 1. And then the next state is 1 if the contribution of the connected genes weighted by the activation or inhibition coefficient is larger than 0. And then so if this sum is larger than 0, it will take value 1. If it is smaller than 0, it will take value 0. And if it is 0, it will stay as it was. Okay. So, and then they follow how this system would evolve over time. And then given a particular pattern, so they start with this pattern. So if you start with this pattern and you follow how it evolves, what every row is a new state. And then they associated the states to the cell cycle state. So in the cell cycle, you have 4 states or 5 states. So G1 growth, and then you have DNA synthesis, G2 growth and preparation for mitosis, and then the cell divides, and then you have 2 new cells that go again into G1 and so on. So depending, each one of these states is associated to a state in the cell cycle. And then what they studied was, it doesn't matter where you start. So each one of these points is a different configuration of these states. So if you start from this state, then you evolve to this one, to this one, to this one, to this one, and at the end, you're at G1. So you see that cells and they have been designed in some way, have been designed by evolution, but they have been designed in some way such that the big states, so these big states, they are very robust. So if there is a mistake in a particular gene, doesn't matter, because as time evolves, it will converge to a state that is well known. Okay, so they are very robust in this way, and that is that gives the title of the paper. So the cell cycle network is robustly designed. So even if there are mistakes in the starting point, still you will go and converge to a state that makes sense. And then instead of having deterministic evolution like this one, so given the state, there is no doubt what the next state will be. This evolution can be stochastic. So you can have a probabilistic state. So the probability of this next state given the current state. And then you have to say again what is this, what are these probabilities, and you have to put numbers and equations on that. But then, for instance, here you have a model on predicting how populations will grow. So the size of the next population at time t plus 1 will be the current one, plus the number of births minus the number of deaths, plus the net migration. And then the number of births is a probabilistic function. So the number of births depends on the total fertility rate, that it will call it like this, of a given country at a given time. And it will be, so the fertility in the next period will be the current fertility plus the decline in the total fertility rate. So this one is, in their model, it is deterministic plus some Gaussian noise. So these births are stochastic, and then they made simulations on how this population will grow, and they did a simulation of how the global population in the world will change over time, and this is their predictions. So these are the points that we have observed. These points that we have observed, we can feed the parameters of the model, and then we can predict in the future. We can use those parameters to predict what how the future will be. And this is the average of those predictions. This one is the average. But then you see that there is an uncertainty associated to it. And this is a very important concept again in models. So when you make a simulation, your simulation, if it is deterministic, there is no doubt. But if it is a stochastic, then it gives you a wider idea. The deterministic part will be this one. So the deterministic prediction will be this. But then everything in nature, it has some uncertainty. You have estimated these parameters. You have uncertainty in those estimates. And then, depending on which is the parameter, there could be different predictions. And this gives you the uncertainty area of those predictions. Different kind of models are these Markov models. So Markov models is a model that you have a sequence of events. So you have so for instance, let's say that we have a number of days, we would have two states. So we have a rainy day or sunny day, and then these are the probabilities of going. So if there is a sunny day today, then tomorrow will be sunny also with probability 0.6, but will be rainy with probability 0.4. If it is rainy, tomorrow will be rainy with probability 0.7 and so on. Okay, then you have a sequence, and then you have, so this is how they evolve, and then you have the probabilities of starting. So do we start with a sunny or with a rainy day? And then there are different probabilities for each one of the state. Okay, so you have a sequence. So you have rainy, 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 then sunny, sunny, sunny. And you can compute what is the probability. So given this sequence, what is the probability of the next state? So the probability of the next state will be given by the probabilities of the previous sequence. So that is in general for all sequences. Markov sequences, they have the property that all this information of the sequence, all this information is condensed into the last symbol. So only the last state has an influence on the next one. So, and if this is true for your sequence, then your sequence or your process is Markovian. Okay, so now you can simulate sequences. So for instance, you have a probability of being sunny or rainy, and then you have the transition matrix. So this is called the prior probability. And then you have the transition matrix, and these are the properties that are the probabilities that we have here. So you can play with them. And once you have your model, you can simulate sequences on them. In this way, so you start, and then randomly you choose any one of these two states with a probability given by this table, by the prior probabilities. And then once you are in a given state, you compute what is the next state, also in a stochastic way, using these transition probabilities. So this is fun for rainy and sunny days. Yeah, and then you can write how this is translated into. So what is the probability transition? So you have the next state is the transition matrix times the previous state, but then you can add an extra variable. The extra variable is the output. So the output is, given this state, I will have some probability, emission probability of observing the output. So now your observation, you don't observe if it is rainy or sunny anymore, but you are at home, and all you see is that someone comes in and this person is either wet or dry. And then you have an emission probability. So you try to estimate what is the hidden state. So this is why it is called hidden Markov models. So you try to estimate what is the hidden state given your observations. So given on the state of that person, you try to estimate what is the probability that you are in a rainy day or in a sunny day. So if it is a rainy day, very likely he will be wet. And if it is a rainy day, with some probability that is non-zero, he may come dry. And the same for the sun. Okay, so now you have how the states evolve over time and how those states are converted into observations. And these hidden Markov models, they are very much used in all kind of genetic analysis. So here is an example of the mutations in cancer. So what you try to see is what is the mutation state that I cannot observe based on some outcome that I can observe. And these hidden Markov models help you to do that. And here is another example, same idea. So how we can estimate what is the hidden state of the genes given the observations that are different expressions. So for instance, we can study these two genes, but there are many others we have not studied. So this is the outcome. And then all the others that we have not studied are the hidden states for us. And then software dealing with these tools, so MATLAB, Python, that are a lot. Then you also have continuous models. So differential equation models, they are still very useful. So you state how your species or concentrations change over time. So this is the derivative, and these are translated from this kind of network. So you have this gene that is helping to overexpress this, or it is promoting this output, and then So for instance, there is a DNA damage, then when there is the DNA damage, it is promoting the expression of REC A, and then REC A promotes this branch, and also it increases this one, and then this one decreases or inhibits these two genes, and so you have a relationship between all those species. And then you have to translate that into a concentration. So let's call it X1, X2, X3. Then you have these inhibitions or repressions, and you have to put, for instance, let's say that we want to say what is X1. X1 is here. Then X1, we want to say that X2 is inhibiting X1, and X4 is promoting X1. So we can do that in this way. So we can say X4 is promoting X1, so the change of X1 over time will be increased by X4, right? Will be decreased by X2. So these inhibitions and promotions, they go into the exponents of the concentration of X2 and X4, and then there is a rate. Okay, and then X1, what is X1? X1 is secreted, so it is eliminated or it is metabolized into something else. And then there is a decrease of X1. So if there is a decrease of X1, then minus X1 to some power. Okay, and the trick here is how do we estimate the powers? So how do we estimate this coefficient, 0.4, 0.2, 0.5, the 10? So all that has to be made on measurements. So you take data that is on X1, X2, and X3, then you see how they evolve over time, and then you try to see which are the coefficients that are compatible with those measurements. And once you have the coefficients, making a simulation is very easy. So you start with some initial conditions. X4 and X5, they have been taken as constants. So we have not included how X4 and X5 evolve over time. And then you simply simulate this. So when you simulate, so for instance, let's say that there is a damage. The damage for us was X6, so X6. And then when there is no damage, it takes value 1. When there is a damage, it takes value 25, let's say. And then X6 will change like that over time. So you see it goes from 0 to, goes from 1 to 25. So 1 plus, during this period, it has increased. I have not plotted in this notebook, but you can do it. Actually, the notebook is in this list of notebooks that I gave you. So when there is a damage, so there is no damage, then there is a damage, and then we see what is happening with the rest of genes. So with the rest of genes, all you have to do is say, okay, X4 and X5, they're constant, X6 follows this pattern, and then I will express the differential equation here. So if you look at these equations and this code, you would see it is the same. So you have 10 X2 to the minus 0.4, X4 to the 0.2, minus X1 to the 0.5, and the same for the other two. And then you have the integration of this differential equation, and this is the solution, how it evolves over time. So you see that when there is a damage, so this model predicts that rec A will increase, so rec A is X3, and that lambda CL will decrease. So that is what is happening. X1 is like this here. So I didn't explain that, and I'm not showing, but lambda CL, the prediction is that it will decrease, and it decreases. So, and this kind of behavior, where you have exponential charging or exponential change of the concentration, it is very typical in many systems. You have seen that also. Have you seen circuits, the charging of capacitors? And so this is the charging and the charging of a capacitor, because the differential equations that you write for a capacitor and for this kind of stuff, they are almost the same. So they share the same structure and they share the same kind of behavior.
And how do they obtain
The coefficients.
yes.
Yeah, so you have to measure. So you have to cause some damage and some DNA damage by ultraviolet radiation or whatever, and then track over time how these proteins and genes, they are changing over time. And then you have a collection. We have it here somehow for in one of the. So you will have many measurements of this kind. So here it was 0.1. In the other case, you will have concentrations. So you have the concentrations, and then from these concentrations, you have to somehow figure out which are the numbers. And I'm talking about a little bit later about that, but you need measurements. Okay, so here we go. Then here you have a little bit of digression of how is it that which are the explanations for these fractional powers? Yeah, so at the end it is not having a 100% efficiency. So there are inefficiencies in the system, and these inefficiencies result into fractional powers. Okay, and then we have inputs. So our input here was a step function. So it was a step function, and then we have different kind of input. So you have step functions or pulses. You have a pulse or an infusion with ramp. So it is increasingly going up to a value, or you have some step function that is also called infusion. So these names, infusion comes from drug delivery. So when you take a drug, can be infused, so it can constantly you have a new dose, or you have a bolus. When you take a pill, what you are taking is having a bolus. Okay, so then gene regulatory networks. So there are many networks that behave in this way. So you have X1 promotes its own activation. So X1 promotes itself, X1 to the 0.4, but then X2 represses X1. And then X1 is secreted or metabolized into something else. So X1 disappears, and X2 is promoted by X1, and X2 is that's it, no? And then X2 is metabolized. Okay, so then you have a feedback network here. So these feedback networks, depending on what is the value of alpha 1, you see that it has very different behaviors. So for alpha 1 that is 0.9, it behaves like this, has some oscillation. And these second order systems in which there are two elements that are involved and that interact on each other, they have the possibility of oscillating. So for instance here, if alpha takes the value 1.02, what we have is an oscillation. And these oscillations are somehow the basis of our day and night behavior. So you are tired by night because there is some biological oscillator that makes you that by night you go to sleep. And that is called the circadian oscillator, and that is a very nice when you travel to somewhere else, then you have to adapt some of the input is light. So here we haven't put light in the equation, but some of the input is light. Somehow you synchronize your oscillator to the light that you see. So when you travel to the US, then you adapt to the time in the US. Then we have seen it a bit with the yeast cell cycling. So it didn't matter where you started, you ended into a given state. So that happens with many dynamic systems. So here is a famous example, this Lorentz equation. This Lorentz equation, what you have is how x depends on y and x, y, the derivative of y on x, y, and z on z, x, and y. And then it doesn't matter where you start, you can track where you go. So you can have this kind of oscillator behaviors. So you start here, and this is your trajectory in the x, y, z space. So then there will be some attractors. So sometimes you may go to an attractor. So once you reach this point, you don't go away from that point, because it does. All these derivatives are zero, and then if the derivatives are zero, that means that there will not be any change. So you can analyze these trajectories in the space, and you can analyze which are the attractors. And attractors, there are some studies that relate attractors and chaotic attractors to cancer. So you have the state given by the activation state of genes, and then some the physiological normal healthy state is some attractor. So if you perturb it a bit, normally you go back to your healthy state again. But if you perturb it too much, you may enter in these kind of chaotic trajectories, and some people are relating chaotic trajectories to cancer. And then the question that we have all the time, how do we estimate the parameters? So all these models, they need parameters, they need numbers. Which are these numbers? And you need measurement. So depending on which are the parameters, so you will have different outputs. So now you can try to do the opposite. So now we will have data, look for the parameters. So let's say that we are interested in the growth of bacteria, and you have what is the growth over different time points with different, so the bacterial growth also depends on the temperature. So you have a reference temperature, and then you measure what is the difference with respect to the reference. And then somehow you measure what is the number of bacteria. So here what we are showing is there is an initial number of bacteria, and there is a final number of bacteria. We are taking the ratio between the two and the logarithm of that. So now you have to put a model. So you say the logarithm of this ratio, and that is my y, so that is what I want to predict, will depend on some constant or some function times time. But this function will depend linearly on the temperature. So that is my model. And then I have to estimate what is beta 0, beta 1 for this model. And a possibility to do that is, okay, you are giving me, so what are my predictors? My predictors is time and the difference in temperature. And then from this time and difference in temperature, I want to predict what is the bacterial growth. So I have some observations on Y. I have some observations on X. So this is what I want to predict. These are my predictors. And there is a functional relationship between the two. And then I can use least squares to look for what is beta 0, beta 1, such that this distance is minimized. for observation. So that is the key for parameter estimation. So you have to define a loss function. So in the loss function in our case here, it is the difference between what we predict and what we observe. And then, but it is this difference can you can evaluate that difference in a quadratic way, like here, in an absolute value, or if you plot this function, it's like this. If you plot this function, it's like this. You can also the problem with quadratic functions is that if the error is too high, then it tends to dominate the sum. And then outliers, they are given very much weight. And then you can do, but otherwise they behave very well here. When you are close to the solution, they behave very well. And then you can try to make a mixture between the two. So you can try to look for a function that locally here, it behaves as the quadratic. So when it is smaller than delta, it behaves as the quadratic. And when it is larger than delta, then it behaves as a absolute value. So this Hoover function is like that. But in general, it is minimized. So look for the beta 0 and beta 1 that minimizes a loss function. So this loss function is this one. So whatever you choose to be the loss function. Okay, so here in this notebook, I gave you some code, and you can explore it, about how to estimate in this particular problem this beta 0, beta 1. So what you can do is, okay, I will explore in a region beta 0, beta 1, and I will explore all possible combinations. So you explore all possible combinations and you evaluate the loss function. So the minimum loss function is here. So this is my beta 0, beta 1. And here is the solution here. So this 0.02 and 0. So the ground truth is 0.02, 0.001. So you see that your sampling was not good enough to discover this 0.001. Okay, so the ground truth, it depends. Beta 1 was this one. Beta 1 was the one on the difference temperature. This data is simulated. And this is the ground truth. It depends a little bit on the difference in temperature, but according to your fitting, it seems that it does not depend. Okay, then, okay, so exploring all possible combinations, it is too expensive. You have to do many calculations. Can we do it something easier? So what you can do is something called gradient descent. Have you seen gradient descent in another course? Yeah, so I will not explain it. So gradient descent, you start anywhere, and then you follow the direction of the gradient. So here, if the function is smoothly behaved, doesn't matter where you start, you will end at the ground truth, and then the ground truth here, so it converges to 0.02, 0.009. The ground truth is 0.001, so much better than before. Okay, then then for some systems, there might be an analytical result. So an analytical result is, so you don't have to iterate. So here we had to explore all possible combinations. Here we started somewhere and then followed the gradient descent. But all these methods were iterative in some way. We can have directly the result. So for instance, if we rearrange this equation and we say, okay, these are my observations, and I write this as an equation system. So I have these predictors, this is my beta 0, beta 1 nouns, and then I have an equation system, and I know what is the least square solution of this equation system. I know the solution is this one. So it is A transpose times A. Now you invert that matrix, A transpose times Y. So you simply do the calculations. Here we have the code for the calculation, and then you have exactly the solution, exactly. And it is affected by noise, but yeah, everyone is affected by noise, so no problem with that. The problem is that many loss functions, they behave like this. So some of them, they have nice global minimum, but many of them, they have local minima, many places. So depending on where you start, you may end up in the correct global minimum or not. It is so minimizing functions, yeah, especially when there is noise, it can be rather cumbersome. There are some other algorithms. All these, they get trapped into local minima. Not this one. This one is the analytical one, goes to a global minimum if it exists. But there are some other algorithms that try to escape from local minima. And here we have a nice example with one of them. This is one of the most known, that is called simulated annealing. So you have, this is the solution, and here is the history of the solution. So you see that it explores for a long time some region that is not very good, but at the end it converges to the global minimum. And the trick is this one. So the trick is, so let's say that this is our landscape. So you start in some place, and then instead of going in the direction of the minimum, you make jumps, and then this jump brings you here. And because this cost function, this is the loss. So because this loss function is smaller than this one, then you accept it. Then you accept it, and then with some probability. So from this one, you try to explore this one, and it is worse. So by gradient descent, you would never accept this solution. But by simulated annealing, you accept it with a given probability. And this probability is as a function of the temperature, and this temperature decreases over time. So you see that temperature decreases over time, and the probability of accepting a new solution, even if it is worse, depends somehow on the temperature. So as it goes down, at the end, you will not make big jumps, but you hope to be in the global minimum. And it is called simulated annealing because it tries to simulate or to mimic the behavior of atoms when you have seen that iron, when you heat the metal, it is it is red, no, it is very much heated. So the atoms are moving a lot, and then they increasingly or decreasingly lower the temperature. So they cool down very slowly, and as they cool down, they find their minimum energy position in the metallic network. So they are trying to simulate the same. There are some other algorithms, genetic algorithms. Have you seen genetic algorithms? Yeah? No? Okay, so they try to mimic how populations evolve, and somehow populations, each one of us have a chromosome, or it has a genetic composition, and then the evolution of the population is based on two operations. One is mutation. So from one generation to the next, there might be point mutations in the genes. And that point mutation may make you more fit. So your loss function is smaller. And the other one is interchange between chromosomes. So sexual reproduction, you take part of the genes of the mother, part of the genes of the father, and then you mix them, and that is a new individual. And then that new individual might be better fitted. So here we can see again how these all this is about having formulating a problem in a way that you can find the parameters. So for instance, here it is, there's a very nice example. So this rat has to find the food, and then you may try to design paths that somehow are better and better until some point. So all this is a genetic algorithm. So at the end, this is the final solution. All the population of all paths somehow combines into. That is very nice. This population of paths. So, and here, this is interesting. So, let's say that you have this population, and then there is a mutation in the code. So actually, this is the snake that wants to eat this rat. So there is a mutation in the path, and then this one will find it. And at the end, all of them, this population will dominate. And this is the solution that it finds. Okay, so at the end, everything is how do I formulate a problem such that I can find the parameters for that. And when I estimate the parameters, I will have some uncertainty. So I will have, we are running out of time, but you will have a value for that parameter, but you also will have an uncertainty for that parameter. And that uncertainty will translate into uncertainty in your predictions. So depending on which are the parameters, if I change them a little bit, my predictions will change a little bit, and that will give me the uncertainty of my predictions. And there is a huge plethora of optimization algorithms. Each one of them exploit the different properties of the problem. And here you have an example. So let's say that I have this observation. So you take this is how a drug that you take is absorbed, and how much drug is left. So at the beginning, everything is left. After a given time, nothing is left because you have absorbed everything. And then you have different observations of it. And then you try to find, you define the amount that is left behaves in this way. And then I need to find what is the parameter K. And then using least squares, you can find the K, the appropriate K. And here we have that problem in a notebook. And then what is the ground truth? What is the value that you have found? So the optimum was this one. So it should be 0.25, and this is what we have found. And all this is called identification. So parameter identification, so system identification is estimating the parameters. And then once you have identified the system, you can analyze it to see how it will behave. And if you know how it will behave, you may try to control it. So I change your input, or I perturb you in some way so that you move to another state. Or I can even try to design my own systems. Here, this is a book. The whole book is available on the internet, and there is another interesting paper that you can. Okay, any question? No? And time for the test. First is, I didn't open the test. Let me. Information.